The date is Professor Will, CBMD PhD. Today, Sunday, July 7, 2019. It is 19:24 and 3 seconds Eastern Standard Time. This is a continuation of my pathophysiology, so let's hop to it here. Discovery reveals prolific ability of Schwann cells to generate myelin. OHSC research could lead to new therapies to heal nervous system disorders. Date July 5, 2019. Source Origin Health and Science University. Summary: scientists have discovered that a special type of cell is much more prolific in generating a protective shelf covering nerve fibers than previously believed. The revelation about Schwann cells raises the possibility of new avenues to treat nerve injuries and various forms of neuropathy. Further research could prove useful in promoting myelin repair in central nervous system disorders such as multiple sclerosis, where damage to myelin slows or blocks electric signals from the brain. This totally overturns the textbook definition of the way Schwann cells work. Two types of cells in the body produce myelin: oligodendrocytes in the brain and spinal cord, and Schwann cells in the rest of the body. Until now, scientists thought that only oligodendrocytes generate multiple myelin shells around axons, a slender projection of a nerve cell that carries electrical signals between cells. The new research reveals that Schwann cells are also capable of spreading myelin across multiple axons. Researchers made the discovery after conducting a genetic screen in zebrafish in the Munk laboratory. They discovered some fish had more myelin than expected. Those fish carried a mutation in a gene called FBXW7. When they knocked out the gene in genetically modified mice, they discovered an unexpected characteristic. Individual Schwann cells began spreading myelin across many axons. It highlights the very plastic potential for these cells. In discovering how Schwann cells generate myelin at the molecular level, the discovery may lead to new gene therapy techniques to repair damaged myelin in peripheral nervous system disorders such as Charcot-Marie tooth disease, a painful inherited form of neuropathy that affects 1 in 2,500 people in the United States. Both Schwann cells and oligodendrocytes arose at the same point in evolutionary history, with the appearance of jaws in the vertebrate lineage. Invertebrates lack myelin, and some, like the modern squid, uses thick axons to quickly transmit signals between neurons. We couldn't have evolved that way, but our spinal cord would be the diameter of a giant sequoia tree. Instead, vertebrate axons evolved myelin to protect axons and speed up signal transmission. To create myelin, Schwann cells evolved to produce it around a signal axon in the peripheral nervous system. Oligodendrocytes, in turn, generated myelin along multiple axons of the more confined environment of the brain and spine, the central nervous system. The real estate is fundamentally different in the central nervous system than in the peripheral nervous system. Research scientists theorize that Schwann cells evolved a mechanism to repair damaged myelin on a cell-by-cell basis, since it would have been common for injuries to occur without necessarily killing the entire organism. Those traits would have been passed down and strengthened through generations of evolution. By contrast, remyelination in the central nervous system tended to be an evolutionary dead end, since few would have survived a severe whack to the brain or spine. There's no selective pressure in repairing myelin damage in the central nervous system because you are probably going to die. However, the discovery published today suggests a new opportunity to heal the brain and spine. Targeting the FBXW7 gene, or downstream pathway molecules, could be a powerful way to promote myelin repair in the central nervous system. Good day, this is Professor Will, CBMD PhD. Today, Sunday, July 7, 2019. It is 19:50 and 40 seconds Eastern Standard Time. This is a continuation of my pathophysiology, so let's hop to it here. Pain signaling in humans more rapid than previously known. Date July 4, 2019. Source Linköping University. Summary: pain signals can travel as fast as touch signals according to a new study. The discovery of a rapid pain signaling system challenges our current understanding of pain. It has until now been believed that nerve signals for pain are always conducted more slowly than those for touch. The latter signals, which allow us to determine where we are being touched, are conducted by nerves that have a fatty shelf of myelin that insulates the nerve. Nerves with a thick layer of myelin conduct the signals more rapidly than unmyelinated nerves. In contrast, the signaling of pain in humans has been thought to be considerably slower and carried out by nerves that have only a thin layer of myelin or none at all. In monkeys and many other mammals, on the other hand, part of the pain signaling system can conduct nerve signals just as fast as the system that signals touch. The scientists speculated whether such a system is also present in humans. The ability to feel pain is vital to our survival, so why should our pain signaling system be so much slower than the system used for touch? and so much slower than it could be. To answer this, the scientists used a technique that allowed them to detect the signals in the nerve fibers from a single nerve cell. They examined 100 healthy volunteers and looked for nerve cells that conducted signals as rapidly as the nerve cells that detect touch, but that had the properties of pain receptors, otherwise known as nociceptors. Pain receptors are characterized by the ability to detect noxious stimuli such as pinching and abrasion of the skin, while not reacting to light touch. The researchers found that 12% of thickly myelinated nerve cells had the same properties as pain receptors, and in these nerve cells the conductive speed was as high as in touch-sensitive nerve cells. The next step of the scientists' research was to determine the function of these ultra-fast pain receptors. By applying short electrical pulses through the measurement electrodes, they could stimulate individual nerve cells. The volunteers described that they experienced sharp or pinprick pain. When we activated an individual nerve cell, it caused a perception of pain. So we concluded that these nerve cells are connected to pain centers in the brain. The research team also investigated patients with various rare neurological conditions. One group of people had, as adults, acquired nerve damage that led to the thickly myelinated nerve fibers being destroyed, while the small fibers were spared. These patients could not detect light touch. The scientists predicted that the loss of myelinated nerve fibers should also affect the rapidly conducting pain system they had identified. It turned out that these people had an impaired ability to experience mechanical pain. Examination of patients with two other rare neurological conditions gave similar results. These results may be highly significant for pain research and for the diagnosis and care of patients with pain. It is becoming evident that thickly myelinated nerve fibers contribute to the experience of pain when it has a mechanical cause. Our results challenge the textbook description of a rapid system for signaling touch and a slower system for signaling pain. We suggest that pain can be signaled just as rapidly as touch. Good day, this is Professor Wills, CBMD PhD. Today is Sunday, July 7, 2019. It's 20:11, 15 seconds Eastern Standard Time. And this is the end of my module on pathophysiology. Hope you enjoyed listening to my reading. Have a great day.